Welcome to our deep dive into anti-drug antibody, ADA assays, essential tools in the development and approval of therapeutic drugs. When we introduce a new drug, our immune system sometimes recognizes it as foreign and produces antibodies against it. ADAs can reduce the effectiveness of a drug or cause adverse reactions. Understanding and detecting these ADAs is crucial for ensuring the safety and efficacy of new therapies. ADA assays are designed to identify and measure the presence of ADAs in biological samples. They're crucial throughout the drug development process, from early research to clinical trials and beyond. Imagine a security checkpoint, the first stage of screening for a big concert. Screening assays in the world of ADAs work similarly. They quickly determine if there are any ADAs present in a sample, like a blood sample from a patient. These assays often use a bridging immunoassay method. If ADAs are present, they bind to the drug on a plate and a labeled molecule detects them. If the label lights up, we know ADAs are present, signaling the need for further investigation. Our screening assay has flagged a potential ADA, like a detective finding a clue. We need to ensure the antibody detected is specifically targeting the drug. Confirmatory assays use competitive inhibition. We add an excess of the drug without the label to the sample. If the signal decreases, it means the ADAs were blocked from binding to the plate because they latched onto the excess drug. This confirms that the ADAs are specific to our drug of interest. We've identified and confirmed the presence of ADAs specific to our drug. Now we want to know the quantity. This is where tighter assays come into play. These assays work by gradually diluting the sample and performing the assay repeatedly. The more we can dilute the sample and still detect the ADA signal, the higher the concentration of ADAs. This gives us a quantitative measure of the ADA levels, helping us understand the potential impact on the drug's efficacy and safety. Not all ADAs are created equal. Some might not cause much trouble, while others can interfere with the drug's ability to work. Neutralizing antibody, NAB, assays help us identify these troublemakers. These assays assess the ability of the ADA to neutralize the drug's biological activity using methods like cell-based functional assays. They measure whether the ADA can prevent the drug from binding to its target or exerting its intended effect. Thank you for joining us in exploring the critical components and challenges of ADA assays. From screening to confirmation, from measuring concentration to assessing neutralizing potential, these assays play a vital role in developing safe and effective therapies. As science advances, so will our methods for detecting and characterizing ADAs. We're constantly developing new technologies to improve the sensitivity, speed, and accuracy of these assays. Together, we can continue to push the boundaries of scientific knowledge and improve human health.